use as monosidase on this, or you take this pool from here and you add a monosidase into it. But you have to look at these. It tells you that you've got other structures there you should be looking at. So it's actually, if you like doing puzzles, it's really quite satisfying to be able to do these kinds of things. Now, you were asking about capillary electrophoresis. You can do, this is human IgG, you can do exactly the same thing with using capillary electrophoresis. You have to have an APCS labeled dextran ladder because you have to have a charge label to do capillary electrophoresis. And we have built into the glycobase about 80 or 90 APTS glucose unit values for use with capillary electrophoresis. So just go to our database. It's probably got the structure you want. If it hasn't, you'll have to work out how to get that structure into your own database. And you can cut and paste these databases too, so you can just personalize it. Here, this is human serum IgG, and you can see here, this is the undigested pool, then this has got sialidase, this has got sialidase and fucosidase, this has got the galactosidase added, and this has got the hexosaminidase added. So you can see, you can go down this exactly the same way, and everything goes back to the trimanosyl core, and the data interpretation is exactly as it is with the, if you use LC. So you can use any good separations technology, you just have to make sure your ladder's right, and you have to check the database because, as you so rightly say, sometimes peaks can shift if you're using a different separations technology. But you'll never really be far wrong if you use the enzymes. Yes?
So the inspecificity of the exoglycosides are improved. I mean, it's not how endonucleases then are a little bit.
Oh no! They're pretty specific, but you have to use them under the right conditions to be sure. Because they will sometimes take a second linkage. They're not, they may much prefer to take a 4-link galactose, but if you press them, and you have high enough concentration, they will eventually take a 3-link. So you have to use them with some sort of understanding of how enzymes work. But actually, if you're doing IgG, they're very robust, and because you've got several checks, you can do mass spectrometry. You also know the pathway that produces these glycans, so you can argue from other places. But the enzymes are just such an important tool, and I think a lot of people now, people didn't like, especially people use a lot of sort of mass spec, are very sort of physical-minded people. They didn't really like it because it's kind of wet bench and it's a bit messy, but one after another, the mass spectrometrists now are just realizing how powerful this is. For example, if you want to look for alpha-gal, you can use coffee bean galactosidase. It's one digest, and you can get the quantitation. If you try and do it by mass spectrometry, you probably won't see it because it's a very small amount, and you don't get quantitation, and it's just messy. So, there are really good reasons to really use these enzymes. And now New England Biolabs have made them. They're so much better quality. You shouldn't have any issues really. I'm just painting. I'm just making sure that you know the snags as well as, I'm not trying not to tell you everything's rosy, just things you need to be aware of. So another thing you certainly will want to do for IgG is looking at the antigenicity. So these alpha-gal, so you see this galactose linked in an alpha linkage to another galactose. So these are pigs, mice, other species than human. And also plant antibodies. The first therapeutic in carrot cells was made by Protalix, by Yoram Tekoa, and they had to check the antigenicity of the product that's coming out of carrot cells. And not all people react to them, but some people do, so the regulators want to know whether you have these residues. And then, of course, this sort of famous evolutionary story that chimps and everything else has N-glycosylaminic acid with this hydroxyl group here. Humans somehow evolved to lose this hydroxyl group, so they have N-acetyluramic acid. And we all have antibodies against N-glycosylaminic acid if you drink milk or you eat beef. So certainly people do have antibodies against these these N-glycolyl acid residues. Whether they actually cause any issues is controversial, but anyway, the regulators don't want to see them because there's a possibility that you might have a reaction to them. So there are quick ways to look for these. You can use the LC or you can use mass spectrometry. So this is mass spectrometric analysis of some work we did with a group in Cuba, looking, and they were growing antibodies in tobacco plants and in alfalfa. And again, you can see here these plants, fucose and xylose residues. And again here, with fucose and xylose residues seen in this glycopeptide analysis. So you can look using mass spec for these epitopes. How quantitative they are, you'd have to establish, but you can certainly confirm that they're there or not detectable. You can use reverse phase to separate glycans. Some people really like this. Roy really likes this. And Roche do, because Roche have an antibody platform where they control fucosylation by putting in a bisecting glutamate. And it's to avoid a patent. But they have lots of bisecting sugars. And the reverse phase will actually separate out bisecting sugars very conveniently. And it basically separates sugars into different groups. So the silated structures here, the high mannose here, the complex type sugars elute out here. Now it's okay. I would say we've looked at it a lot of times and we don't use it because it's very heat sensitive. So if you have a pre-column heater, you get a different separation order from if you don't have a pre-column heater, and it's just much less robust for us anyway. So we had a long project with Roche. We worked out how they could use it because for them it's a good idea, but for a first approach, I wouldn't use reverse phase. You can also, sorry.
In reverse phase, which one is your mold phase? Because in Kylie, we use hydrophilicity column.
Yes, no, this is hydrophobic. I don't really know. So probably you start off with acetonitrile and dilute it out. But that's another problem actually. You use a very, very shallow gradient, and so it's not very reproducible. And it's because you've got the gradient is so shallow, and you get diffusion, and so I think this has specific uses, and where you like Roche, you need it. It's extremely good. But if you're just starting, this is not your number one technology to use. Use CE, use LC, but I wouldn't use this unless you really, it has features that are really important to you. And if you're going to look into the future, this is ion mobility. And this is like a new technology that people are currently trying to predict where the glycans will run and just explore how powerful it's going to be. But if you have a synapt with an ion mobility tube on the top, this is like chromatography in the gas phase. Basically, it's gas chromatography, but it's much more sophisticated than the old gas chromatography. So it will separate different charges of glycans, for example. So if you're a mass spec buff, this is a technique of the future, I suppose. So now I just show you how you can use some of this technology. So I talked yesterday about glycoengineering CHO cells and other cells for that matter to get carbohydrate-defined products. So these genome editing tools that we have in Singapore, those zinc finger, Talon, and now CRISPR, which is probably what everybody's going to use. And so what Song Sui and Pei Ching were able to do was to develop these cell lines that were producing fucose-free IgG, sialic acid-free, man-terminated sugars, highly silated EPO, and there was much less heterogeneity. And obviously in bioprocessing, if you want homogeneous glycoforms, then this is very important. And there's this publication, and last week I think a second publication came out from Zhang and Pei Ching and from my group. So I showed you this picture before. This is the anti-HER2 antibody, and this is data that came out from Singapore, from BTI. So this is the intact IgG glycoform study. And so we then, with these innovator, we looked at the innovator product, trastuzumab, and we then looked at the glyco-engineered version, which had no fucose, which was engineered to have more effective ADCC. So here you can see in the innovator product, a lot of this material has caulic fucose. And if you compare that with the material that's come from these, the knockouts for GDP fucose, you can't detect fucosylated structures at all. So this is knocking out the transporter, and obviously it's very powerful in controlling fucosylation. And you can use the exoglycosylase arrays as an orthogonal way of confirming this. And again, you can see that the fucosylated structure is when you get to this stage and you use the fucosidase, you can see that nothing has changed between this profile and that profile. So there's no, and if you blow it up, you still can't see it. So there's no detectable fucose after this engineering out the transporter. Another reason that you want to use analytics, apart from understanding whether your gene editing has worked, is to use it for clonal selection. So these are a selection of clones that we were testing for Herceptin. So this is the analysis of G0, G1F, and G2F, which are the critical features that we need to report. So here you can see Humira, and this is what we're aiming to replicate. So you can see most of these clones are nothing like Humira in terms of the relative amounts of these different glycans. The only one that comes even close is this one here. So what this is helping us to do is to say, well, it's not worth looking at any of these clones because none of them can produce glycosylation like Humira, but we might persist with this one because at least this is reasonably close. So this is an important thing to be able to do when you're developing cell lines. And also glycosylation sensitive to the bioprocess. So here's an example where we took, we were making an antibody and we added particular feed, and then we put in a food, the same feed, and we put in a hydrolysate, and we put in different levels of hydrolysate, and we changed the temperature. And then we monitored the glycans, the G0, G1, the whatever, high mannose glycans, fucosylated glycans, G0, G1, and G2, under each of these bioprocessing conditions. And here's Humira, so that's what we're trying to make it look like. And again, you can see that few of these really are very close. But on the other hand, this one probably isn't bad. So as we've gone along this progression, we're getting closer to the innovator product, but we still haven't really got there. Also during production, it's really important to monitor the production. So this is looking at increases in this MAN5 structure during different days. So these are different days of the production. So you can see as you get further along the production schedule, you get more of this MAN5 structure. And as I said, this is probably because the cells are running out of the manosidase, which is needed to cleave the mannose back to be able to make a complex glycan. So you don't want to get lots of this material in your final product. So you may decide, this is enough, we'll stop it at day 10, or you may be able to vary the conditions, change the dissolved oxygen, the media constituents. So you may, if you know this information, you may be able to do something about it. So monitoring production is really important. So all I've shown you now, most of what I've shown you has been LC. So the other part of the orthogonal technologies that we use is mass spectrometry. So here's the LC part we've just looked at. If you have an online mass spec, so Waters have this complete system, then the sample will go through here, but it'll split, so half of it will go into the mass spectrometer, so that you can actually get a mass spec composition for all of your peaks that you have on the LC. And that kind of workflow enables you to support your glucose unit analysis with mass spec intensity extracted ion chromatograms, mass spectrum fragmentation if you need to. Now, you don't have to do it online, so we still don't have it online in neither. We just take the pool of glycans and we do the mass spectrometry on a different instrument, and we use that. So you don't have to have it online. And you can do something as simple as having a MALDI, just doing a composition, matching up the compositions. So this is the Rolls Royce, but you can get on quite well with the Mini. The other thing is that Waters and there's a Waters person going to give a talk next, so I think they're going to go into this probably. But Waters have developed a very powerful label that replaces 2AB. Now, we developed one as well, which is not the same, and for research purposes it's fine, but it's not QC'd for regulated environments. This one is. It's expensive, but if you're having to work in a regulated environment, it's worth it, I think. But the really good thing about it is that it's very quick label, which is not particularly novel, but it is very sensitive in the mass spec. So the problem that we've always had is that the LC is highly sensitive, so you get beautiful LC profiles. Then you put it on the mass spec and you don't see anything because the sensitivity of the mass spec is so poor. So this label has been specifically designed to give you very high mass spec sensitivity. So here you can see this is the rapid fluor label compared with the fast 2AB label, the instant 2AB, compared with the 2AB that we conventionally use, and procainamide, which is another label that people like to use. So this new label is very, I think, twice as sensitive in the fluorescence compared with anything else. But if you look at the mass spec, the difference between its best competitor, which is procainamide, and the new fluorophore is enormous. So it is 100 to 1,000 times more sensitive in the mass spec. It's really helpful. So here, if you compare the rapid fluor with the instant 2AB, it's twice as sensitive in the fluorescence detection and 1,000 times more sensitive in mass spectrometry. So this is a really helpful addition. And the reason it does this is because it shifts the glycans to higher charge states. So you see more charge states, and you can get more information that enables you to get more sensitivity. And it's also very convenient across a whole range of different glycans. So this is just a compilation showing that it works for biantennary structures, for high nanostructures, and for large complex silated structures. And so that's the fluorescence and mass spec response. And if you look here at the profile, this is just saying that it's got a very big dynamic range, so you can identify this peak, and you can also identify a very small peak down here. So as I say, I think the Waters rep, obviously isn't just here, it will give you more information about this. So that's IgG. So I think I could talk for ages about IgG, but I just wanted to get on to something else. So if you had any questions about IgG, maybe this is the moment. Yeah.
Pauline, when you have set one condition and you have a profile, how reproducible is that over time?
Robust, really robust. I mean, we can go back. If you use the same conditions, you get the same profile. I mean, we've done it many, many times. And the other thing that we always do, we run the dextran ladder about every 10 runs. We run the dextran ladder just to make sure that the plate count is staying the same, and there's no shift in anything. And I suppose the only thing that would change things would be if you just use your column until it was dying, and the resolution was very poor. Then obviously that's not a good idea. But that's not the fault of the technology. That's the wearing out of the column.
And I'm thinking more on the culture side. How reproducible is
the cultures?
The glycosylation profile that you get when you use the same culture conditions over and over again?
Oh, I see. Yeah, well, I don't have a lot of data on that because we don't have those samples. But whenever we've done it, it makes sense. So when we did the 20-day study, it wasn't, the amounts were all similar, and you could just see the rise of one peak and the fall of something else that matched it. So, throughout a culture, it looks stable. If you do the same culture over and over again, I think you'd have to go to a pharmaceutical company, because the most we see really are the end products. They don't give us all the different culture stages. So I don't know how stable the cultures are.
Si alguien quiere preguntar en español, con mucho gusto traducimos, ¿eh? No se inhiban por eso. And another question is about the purification steps that you do before analyzing this protein. You do only protein A, or do you use the whole purification train that is usually used by?
We can do. If we are looking at an end product, we would do that. But no, we usually just use protein A, because we know that we can wash out host cell proteins, and we can check there's nothing significant there except for IgG. Or we could use hydroxyapatite, like Pete Gagnon, for example. There are different technologies for pulling out IgG. But we were trying to design something that you could use quickly at line. And
my third question is, have you seen any condition that results in silylated glycans in antibodies?
Results in desilated or silated? Yes. You can promote them, mainly by gene editing. So I've seen data where the sialic acids have been removed, but not where they've been increased. So I guess if you put in, if you look at Mike Butler, his work, he's probably done that, or Roy probably has done that, but certainly Mike would have looked at ways to increase silation. So.
Thank you.
But I think this is all going to be superseded, personally, because I think the CRISPR is so powerful that, hi.
And in the study that you analyze the profile of glycan profile, did you analyze the activity, exoglycosylase activity?
No, we do it when we get a new batch. When we get a new batch of enzyme, we test it on the IgG profile or a human serum profile sometimes. We just take release glycans that we've analysed many times, and then we check the enzymes behave as we expect them to. And so we start off using the number of units that the company says is in the bottle, but if it doesn't work very well, then we have to use more enzyme, or we would optimise it. But I mean, on the whole, they're pretty good. I mean, as long as you don't go outside the sell-by date, then you're obviously it's at your own risk. It may be fine, but mostly we just run a couple of checks just because we like to know what the enzyme digest looks like with that particular enzyme.
And the lead gene that codifies alpha-2,3 cell transference in CHO cells do not affect the physiologic state of the cell. I mean, why does CHO cell have alpha-2,3 cell transference?
It's a very interesting question. There is a 2,6 gene there, but it's just not expressed. It's a silent gene. So at one point we had a big effort to try and wake this gene up because we thought it would be really good, but we were trying to make the ligand for PGSL1, which has 2,6-linked sialic acid, so we were trying to make it in CHO cells. But we never could wake up that gene. The only way we got 2,6-linked was by editing in the gene for 2,6-linked sialic acid into the CHO cell genome, the human one. So I don't know why it only puts on 2,3, but if you could wake up 2,6, so you'd have the Nature paper. Yeah. So.
Which is a good threshold to start to quantitate peaks? Because in a profile there are many peaks, and for example in IgGs, 4 peaks are the major structures, but in mice, in rodent, there are many other peaks in minimal amounts. Which is the last peak you
well, we would determine any peak we can see, and we would normally go down to 0.05% of the total glycans. That's what we can analyse quantitatively with confidence. But so if you're first looking at something for the first time, you probably want to do that. Well, we would do that. But if that's not a critical feature, those peaks are not critical features, and you don't need to know those structures every time, then you might just decide to go for the top 20 or what. It's a judgment how many you would look at. But if we can see it, we do analyse it. Yeah, and that's why the LC, you can often get peaks on the LC that you don't see on the mass spec, because the LC, until this new label came along, and we haven't really tested it very thoroughly yet, but you would see small peaks on the LC because the signal to noise is huge, and also you can go down into the baseline and you still have very good signal to noise, so you can be confident about very small peaks. But I think it's a kind of a judgment, really, whether you need to know all those small peaks but if you don't, you need to point out in your report that you saw these peaks and you didn't further analyse them, because they might be important. Small levels of an antigenic peptide can be very important in some people who are hypersensitive.
In terms of glycosylases, you said it's important purity of the glycosylases. Do you know a good source of the glycosylases?
New England, well, I'm promoting a company here, but we had a collaboration with them. So New England Biolabs have bought this whole set. They call it the NIBUT9. The 9 major enzymes that we use, we worked with them to make those enzymes recombinantly, because a number of them, like almond milk glucosidase, we and everybody made it from almond meal, and we know there were other enzymes co-purifying with it. So they got the DNA, and as I say, they had to grow an almond tree to get intact DNA, and then they made the enzyme recombinantly, and they did it for all these 9 enzymes, so that you don't see any co-purifying contaminants anymore, and they're properly the activities are properly tested. So that's what we use. But as I say, it was born out of our frustration of getting enzymes that sometimes didn't work very well, sometimes had a lot of other stuff in them. So I think it'll make the other manufacturers clean up their act a bit too. Originally, all these enzymes were made in Oxford by the Searle Group. So in the Glycobiology Institute, we had a group of enzymologists from Searle, a pharmaceutical company, and they made all these enzymes. And they were fantastic enzymologists. They looked at all their activities, they worked out all the activity, all the number of units that you needed to use to get these activities. And then the Glycobiology Institute spun out Oxford Glycosciences, which was far ahead of its time. I mean, its enzymes were squeaky clean. And then that company was bought by Celtec. Celtec was bought by Glico. Glico was bought by Prozyme. And every time this enzymology was bought by another company, it got more and more sloppy. So I think now we're back to where Oxford Glycosciences were. Well, we're better because they're recombinant.
And the last one. I've seen papers on glycoengineering where they show silated glycans or IgGs or diglycosylated glycans, but the major structures are GO, G0, G1, G2. Why we don't have high G2?
Oh, well, it's because of the structure of IgG. So this is what I was saying, is it's worth spending some time just understanding the structure of IgG. So maybe I can just do it with hand waving. But so you have the CH2 domains. You have 297 here, which is at the back of the molecule. You have 297 at the front of the molecule. The sugars come out into the cavity, and they have low affinity interactions between the arms of the sugars. Right? At the same time, the sugars are actually interacting with the protein surface. So there are 96 interactions of those sugars with the protein surface. And this was done in the 1980s by Padland, so very well established. And some of those are very weak. They're just hydrogen bonds or van der Waals forces. But when you add them all together, they actually, and you also add in the final galactose residue actually sits in a pocket in the protein. So once the galactose residue goes on, the sugar is pretty much pinned against the protein by that galactose fitting into the pocket. So once that goes on, it becomes, in order to get acylic acid onto that galactose, it's got to come out of the pocket, which is unfavourable. It also has to see the silyl transferase and the CMP sialic acid simultaneously because it's a ternary interaction, and it's in a confined space, so it's very unfavourable. So that's why you see silation in the FAB where the sugars are free, but in the FC you just see very little. So if you look at human IgG, which has 20% of FAB glycans, you see silated structures, but they're nearly all from the FAB, right? So, but then the other problem is that once you've got one galactose in the pocket, the sugar is just not very free to move. So actually getting galactose on the other arm becomes difficult as well. And the two arms are preferentially galactosylated. So the 1,6 arm, that gets galactosylated first. It sits in the pocket, and then to get it on the 1,3 arm is really very difficult. If you look at the molecular model, you can see it just isn't accessible. So that's why you get not so much G2, and you get G1 distributed on both arms, and you get G0. Now, those proportions are different for, if you have rheumatoid arthritis, you have a lot of G0, and you have some G1 and some G2. But, a lot of diseases and a lot of bioprocesses actually favour one particular glycan, partly because of the accessibility problem, but also because of the levels of the galactosyl transferase. So IgG is very unusual because it has the sugars buried in the cavity. So the amount of the activity of the enzyme and the amount of it are absolutely crucial for IgG, for the FC. So if you look at the FAB, it's no problem because if the level of galactosyl transferase goes down a bit, it doesn't matter. It's still got plenty. But in the FC, it's actually really critical. So if you have a disease where there's less expression of the galactosyl transferase, it will immediately show on the IgG sugars because you're just below the threshold that you need for the enzyme transfer to happen. So as I was saying, honestly, it's worth looking at a few reviews on IgG structure and just sort of understand how the sugars actually relate, because they're different from a protein like this where the sugars are all on the outside. Like there's no restriction. There is restriction from the 3D structure, but nothing like there is buried in these CH2 domains. So that's why you never see it's very difficult to see tri-antennary structures on IgG or tetra-antennaries. You get other proteins coming out of B cells, and they're quite happy with tri-antennary structures, but you don't see it on IgG. And
in that terms, do you think the galactosylation can be manipulated or not?
Yes, you can express more copies of the galactosyl transferase, and you can force it to become more galactosylated, and have more of the whatever it is, UDP galactose.
I didn't see any paper about how to increase the level of G2, for example.
No, well, people have tried that. They try to add it on. I saw a paper came out the other day where somebody was doing silation, actually, and they were post-expression putting on galactose and sialic acid. So, you can make your IgG, and then you can try to add galactose and add sialic acid. And, I mean, you can make it work, but it's not... I've tried it. It's not exactly trivial, but if you get the right conditions, it's obviously it can be done. So, but IgG is kind of frustrating, really, because years ago we wanted to look at individual glycoforms of IgG, so you can make G0. It's really easy. You just use the silidase and galactosidase on the whole molecule, and it works. But to get G2, you can't do it like that. You have to set up to isolate that particular population on lectins, or and then the lectins don't get in for the same reason. There's not much room; they don't always get in. So, yeah, it's a challenging molecule, but it's an interesting. It's always something new with IgG. Okay. Yeah.
I would like to know if I'm so curious. I would like to know if the biopharmaceutical industry use mass spectrometry as a technique. As a gold standard technique for analyze glycosylation in each state of the lab production.
Well, they do. There's a few, I know Roche They use LC, and they use 2AB labelled LC. If you look, Dietmar Reusch did his PhD on. He studied all kinds of every glycoanalytical process that there was in order to decide which to use in Roche, and they ended up with the reverse phase, and their bog standard one is what we use, 2AB. And one of the reasons, the main reason they use it is because it's actually analytically more information. But the other reason is if they do mass spectrometry, they have to take the sample from the production lab, send it over half a mile over the other side of the campus, put it in a queue, probably get the answer back 3 days later. Where if they have LC, they can have it in the production lab, and they can just run their sample straight away. So most companies, I would say, are using 2AB labelled normal phase and helic separations as their bog standard way of doing it. But companies also use mass spectrometry. They will use it as well. Some companies, they use primarily mass spectrometry, although they're getting less. I mean, where mass spectrometry is really powerful is when you want really detailed glycan analysis. So that tetraantenary glycan there, if you didn't have sialic acid on one of those arms, I'll show you if I have time, you could find out which arm it was missing from with mass spectrometry. You couldn't do it with LC. So detailed glycan analysis, it needs very powerful mass spectrometry. So I would say people do use it. And actually we're just doing some tests with Manfred Werher in Leiden. I mean, provided you don't mix types of glycans, the MALDI is quite good at quantifying actually. It's only when you have nanostructures which fly better, so they're overrepresented, and you're looking at sialated structures which probably lose sialic acid in the experiment, and you're trying to monitor them both in the same run. That's when you have problems with mass spectrometry. But if you're only going to look at nanostructures, you will always get that'll be okay. You'll get good quantitation. And the other issue, see, when we do LC, we always talk about relative quantitation. So we take all the area and all the peaks, that's 100%. And then we say this glycan represents 58% of the glycans in the total profile. So the mass spectrometrists always want to talk about absolute quantitation. And that is very difficult. I mean, how do you know your standard is accurately weighed? It might have, you don't know it's only sugars. I mean, I saw some data the other day where somebody thought they had 98% pure sugar, had 5% of DNA in because they hadn't looked for DNA. So actually knowing, that when you weigh something out and knowing how much water there is there, I mean, that in itself is like really challenging if you're talking about making accurate standards. So, and also with the mass spec, if everything flies with more or less efficiency, you've got to have a standard for every sugar in your profile. So think of EPO with at least 70 glycans. You're going to have 70 mass spec standards running, So people then try and do isotopic labeling to sort of measure a standard against the particular glycan. So you measure the amount of your standard isotopically labeled, and then you can quantify your, such. But I think finding out absolute quantitation is really, really challenging, and that is people trying it, but it's going to take a while to get it.
Is your group tending to produce biosimilars?
No, my group doesn't produce biosimilars. I mean, I work with groups that do, like in A-STAR, we work with the Animal Cell Technology Group, and they make biosimilars, and some of the data that I'm showing you from A-STAR is their data from biosimilars. But my group, we specialise in the glycoanalytics, and obviously I've got people who do cell. Well, I mean, my group's very broad, if you see what I mean, but the main focus is being the experts in the glycose.
If you want to choose a lines that produce different quantities of the same recombinant antibody, do you expect use change in glycosylation profile?
When you scale up, did you say? I didn't quite hear.
So if you have two chosen lines that produce the same recombinant antibody.
If you have two...
Cell lines.
Oh, cell lines, sorry, yeah. Yeah.
Do you expect a huge change in glycosylation profile of the antibody from?
Yes, huge.
Yes, because
Yeah.
I mean, there are different quantities of the antibody that are being processed in the classical secretion pathway. So, I mean,
I'll show you with EPO because I've got some data, but the same applies to any recombinant protein. The cell is the first determinant of the glycosylation pathway.
But I mean, it's the same. It's the same just a line under the same culture conditions, but they produce different quantities. I mean, they have different productivity. So
different products. Did you say the same CHO cell line but different products?
No, the same. They secrete the same antibody, but they have different productivity.
Oh, different.
The rate.
Oh, the productivity. It's the same CHO cell line.
Yes.
There's different productivity.
They secrete the.
Well, the media. Yeah, I mean, the media will affect the productivity. All sorts of things will. I mean, you can change the productivity of the cell line. So.
But the question is if the glycosylation pattern or profile change, the cell line is the same culture conditions, but the productivity is different. I think
well, they won't be the same culture conditions. Something will be different, and they'll be maybe pushing more through, and then the glycosylation pathway won't handle it in the same way. So.
No, I mean you have two different forms of one cell lines that produce the same antibody. So they have different productivity. They have basically different quantities of the antibody. Do you expect that a huge change in the glycosylation profile of this product?
Well, actually, do you remember the slide I showed you on clones? Yes. They're all clones from the same CHO cell, right? The parent, but they are very different, and they're under different culture conditions, and just the way the cells are picked. You know, some of them are just different. So I showed you. Did that? Fine. Do this. So these are different clones, right? So you can see the glycosylation is different. And then the next one is the bioprocess. So if you change the feed, if you start putting in Marmite or whatever your hydrolysate is, your yeast hydrolysate, you change the glycosylation. So this is why it's so difficult to make a biosimilar, because you know how it's like a rheostat. You know, how do you control it? So. I mean, the glycan analysis to me is really straightforward compared with the cell culture. And it's easier to make an innovator product than it is to make a biosimilar. I mean. And you don't want to buy better either, so I think it's a big challenge. And the regulators have to take this into account, otherwise these drugs are going to be so expensive, nobody will be able to afford them anyway. So I think they have to come to terms with this somehow. Okay.
Last question.
Yeah.
Why in the profile of the monoclonal antibodies we find high amounts of M5, but not of M6, 7, 8, or 9? Because the enzymes that cleave back, they can reach the MAN6, 7, it's the monocytose that's close, takes back the final, because you probably have to have a higher concentration to take them off, to get back to the precursor for the complex type glycans. And the last one comes off once one arm is becoming complex. You have to have the glucnac on, and then the last mannose comes off. So I think you have to have a higher concentration to do that, of monocytose. So, yeah. Okay. Okay. So should we go on to EPO? Because this is like a different challenge, really. So this is erythropoietin. It's a very good model because if you express erythropoietin in different cells, you get very different glycans, and I'll show you that in a minute. Glycosylation is cell-specific,
So as we just said, it depends on the particular set of processing enzymes in a cell and their levels of expression. It's protein specific, so if you express two proteins in the same cells, they'll have different glycosylation, because the protein itself can determine to some extent the type of glycosylation that it puts on its proteins. Glycosylation is site specific because of crowding by the protein at any one site, or the effect that attachment of one sugar has on the processing of a sugar next to it. So the site is specific, and also the process. So if you change the process, as we were just saying, you can change the glycosylation. So erythropoietin is used for many things, including treating anemia and cancer. And it has three sites which preponderantly have different, they have overlapping sugar structures, but this one is practically all the tetrasylated glycans. These, you can get different types of glycans at these two sites, although in a pharmaceutical product you really want tetra and ternary fully silated glycans everywhere. And erythropoietin also has an O-link structure which is attached here, and this is very simple in CHO cells. It can only put on galnet, galactose, and then plus or minus the sialic acids. So the glycans, the reason the glycans are really important is because they extend the half-life of the protein. So if you have sialic acids on here, it can't bind to the acylo-glycoprotein receptor, and so its half-life is extended from something like 3 minutes to 3 hours. So the glycosylation is a critical feature. They also stabilize and protect the protein. So if you put a sugar on a protein, you get, and you do proton exchange experiments, you'll find that the protein breathing is damped down all over the protein. It's not just around the glycosylation site. It has a general stabilizing effect on the whole protein. And it also protects the protein from proteases, because these sugars are moving around the N-glycosidic linkage, so they cover a massive area and protect it from protease digestion. And they can also modulate the activity, and as we know, they can be antigenic. So there are lots of different EPOs on the market. This is erythropoietin alpha. This is darbepoetin alpha. This is Aranesp. It has additional glycosylation sites. So they have like 3 glycosylation sites and then two additional sites which were engineered in to increase the half-life. So erythropoietin, it's a 36 kilodalton protein. It's a cytokine. It binds to its receptor. It stimulates an increased production of red blood cells. It has naturally three N-linked glycosylation sites, one O-linked, and 40% of its molecular weight is glycans, and high levels increase the half-life. So this is how the EPO world looks. The first generation was EPOX. The next generation included neorecormin. Then EPO was produced in a human cell line by Shire, that's Dynepo, and then EPOMAX, darbepoetin, with 5 N-linked glycosylation sites, that's Aranesp, and then some people pegylated. So there are lots of types of EPO and erythrocyte stimulating molecules on the market. So this is just a few that we looked at. These are 4 on the market. This is a human cell line, and these are 3 CHO cell lines. So this is what I was saying about, you just sometimes just want a quick 1D separation of glycans by a single technique, just to give a signature or a single snapshot in order to be able to compare the glycan profiles of your product with other innovator products. And this is just a glycan release 2AB label and run on a UPLC column. And you can see it gives you a lot of information. It tells you the human cell line product is really different from all the CHO cells, and the CHO cells are different from each other in the way they process the glycans as well. This is LC, but you could do it by NMR, capillary electrophoresis, you could use leptin chips, you could use mass spec, you can use a whole range of technologies. But the higher the resolution, the more confidence you're going to have in your ability to distinguish one glycan profile from another. So with EPO, because charge is very important, we first of all do a weak anion exchange chromatography. So this is released glycans, 2AB labeled, separated on a weak anion exchange column to show you the non-silated, di, tri, and tetrasilated material. So just a word of warning here. If you have, there's no neutral glycans here, but if you have neutral glycans on weak anion exchange, you should check that they're actually glycans, because within the neutral structures you sometimes get junk there. So you should always take that pool and run it on the LC to get rid of any other non-glycan things that have eluted in that peak. But with EPO it's not a problem, because you can divide and you can quantify the relative amounts of these different charge states. So if we do that for these different EPOs that we looked at, you can see the charge states look very different from each other to begin with, and you can quantify all that. So we can look at the percentage of monosilated di and tri and tetrasilated glycans, and we can also look at other silated structures that we found, like disilated structures, for example. And you can see here the differences. So 62.3% of dinepo is tetrasilated, and EPEX there's 53%. So just from that wax profile you can get a lot of information.
All of them are innovators, because they have different.
Yes, they're all innovators, I think. Yes. I think they are, because I don't think any biosimilars have really been allowed yet. The FDA still hasn't approved any, so these must all be, they must have some differences in that make them innovator products. Yeah, so you need to watch out for things that you might not be expecting. So it's why you need to do full analysis. But there's other technologies that are coming on stream. This is actually a chip from Agilent. So we had a thought leader when Oscar did this work. So we took these the anion exchange pools and we put them onto the chip, and then we separated them on the chip in wax, and then we bumped off these different fractions onto an HPLC matrix, a normal phase matrix, all on the chip. So this is twice the size of a bank card. They're very minute amounts of material. And then, so we had some switching valves to do that. And then eventually we went into mass spectrometry, and they're not very good mass spec profiles because we had a not very good mass spectrometer. But essentially these separation technologies on the chip actually work pretty well. So there are still a number of issues, like the fragile nature of the chips and things, but in the long run these are going to be very powerful, I think, in miniaturizing glycan analysis. So one way or another you separate your structures according to charge, and then you take each of these pools and you run them on a Hillock column. So this is the total release glycan pool of the EPOS glycans. And then when you take them monosilated and run it out, it looks like this. This is the di, this is the tri, and this is the tetra. Now the reason this is so useful is that we know sialic acids are labile, and especially in the mass spectrometer. So you can even desilate this material, and it won't matter because you know that originally it was monosilated. So if you lose sialic acid, it doesn't matter. And the same with the disilated. If you analyse all these profiles separately, you'll be able to piece back together the silation level. So when you do this separation, these are the different silated fractions, and this is the whole pool. So you should always check that when you overlay the silated fractions, you get back the profile that you started with, to make sure that you haven't lost anything. And then you can take, for example, this is the glycan pool from dinepo. You can do all these enzyme digests, and you can analyze these glycans just like I showed you before. And one of the ones that you come up with is this tetra-antennary tetrasilated glycan. And that you need an orthogonal technology to identify that glycan, and also other very large structures like this tri-antennary glycan, which is present in dinepo. So this is just showing you that this is the mass spec profile of the undigested material. Just here is the peak that corresponds to the composition of this glycan. And here we've used the enzymes to cleave back these peaks and to follow it in the mass spectrometry. So if you really want to use mass spectrometry, that's great, and you can use these enzyme digests just as you would in LC to improve the understanding that you have of the peaks in these different pools. So instead of an LC profile here and looking at the enzyme digests, you're going from a mass spec profile to a mass spec profile. So this is really useful. And you can pull out a particular ion that you're interested in, this very complex ion here, and you can do fragmentation, and you can identify where the sugars are, and you can get information that you can never get from LC. So this is why mass spec is so powerful. Another thing you might want to do is report the core glycan structures. So this is the number of antennae that each product has. So and also the levels of this core glycosylated tetraantennary structures. So again, here we've reduced everything using the enzymes, and then we've quantified the different antennae that we have in the glycosylation levels. So you can easily see that this FA2 is very low in dinepo, it's higher in RNS, and so on. There are other things that you need to be aware of, and that is in cell culture you often get acetylation of sialic acids. It's an artifact of the cell culture, but this is again why you need mass spec, because you don't see this on LC, because the acetylated sialic acids come off with the enzymes. If you put them on the mass spec, you'll see them because they obviously have different masses. So again, these the British standard EPO that they will supply from the NIST, from the Standards Agency. This is what it looks like, but it's very odd because it's got these lactosamine repeats here. So, you know, nothing is, you can't take anything for granted when you're doing glycan analysis. You really have to be sure that you're not just jumping to conclusions. You must check, because some of these things, it could be a biantennary structure, it could be a triantennary structure, or it could be a biantennary structure with a bisecting glucnac, and that would look the same on, that would have the same mass, but it would probably run differently on the LC. So just orthogonal confirmation is just very important. So if we look at how we would get a quick result on these antigenic epitopes, this is the mass spec. It's very easy. You don't get quantitative data, but you can see these diagnostic ions. So if you have N-glycolyl acid, the diagnostic fragment is at 306. If you have sialic acid there, and acetyl sialic acid, yeah, the diagnostic ion is at 290. So just a simple mass spec will enable you to demonstrate that those, that those ions are there. I think there's a mistake. Anyway, the other way of doing it is by LC. So this is a kit from Ludger, which has a series of standard N-acetyl and glycolyl acid standards. So you run your profile of your release glycans, and then you can match these to the Ludger standards. And you can see in here, for example, you've got N-glycolyl acid, you've also got another species of N-glycolyl acid. So you can do it by LC, you can do it by mass spectrometry. And then when it comes to site analysis, so first of all, you want to know which sites are occupied, and you also want to know which sugars are at which site. Now, this is really state-of-the-art stuff. It's really not easy to do this. You need to get an enzyme that will cleave these sites from the protein so that they're all independent. So you've only got asparagine 24 on sugars on this peptide, and you've only got asparagine 38 sugars on this one, and so on. And since these are quite difficult to separate, we usually use Glicociencia endoproteinase. And then we go into biopharma links, and we can find the glycopeptides. So on the LC that we use after we've done the tryptic digest, the glycopeptides tend to separate in the same region of the curve of the profile